talk about proteins in the system, we talk about initiation, the elongation process, and the germination process of prokaryotic cells. I want to say a few things about eukaryotic cells, but mostly about initiation. Elongation and termination are pretty similar. So I just want to tell you about initiation. Remember that messenger RNA, as I said before, at the 5' end and the 5' cap, which is setting up a G cap on the end, so there's no phosphate, no free phosphate, which somewhat protects it from the digestion with 5' and 3', also indicates the go in this direction. We also said that the 3' end has the polyA tail on the end, the polyA tail from 200 to 250 adenylate residues on the end of the RNA. They also have an AUG start sequence that starts the translation to proteins. And remember, the prokaryotic cell, ribosome is pretty much, or at least the small subunit with the ternary complex, that 30S initiation complex, address the messenger RNA at the 5' end, and then begin to walk down looking for an AUG. But it didn't look just the first AUG, for that special AUG that was preceded by that Shine-Hill-Garner sequence, that AGGA sequence, which is complementary to the 3' end, 16S RNA that's in that small subunit. It's part of that 30S complex that's walking down. And when that complementarity is realized, that indicates that the AUG that's at that position is a start. So that ribosome will march on down here until it finds the right AUG. Eukaryotes are different. The first one is different. Ribosomes do bind at the 5' end, but they bind as a result of proteins and complexes that form around this cap. And I'll show you that complex in a minute. There are several initiation factors that bind to the cap. They then, once they bind, they move on down to the very first AUG. There is no Shine-Hill-Garner sequence or analogous sequence in eukaryotes. Rather, the ribosome moves on down to the first AUG that's there. So in this untranslated region, the first AUG is the start of the bond. So how do ribosomes initiate here? There aren't special initiation factors that are different or that are additional. I showed you this figure here, and I showed you that after that 30S complex with the initiation factors 1 and 3, which keep it separate, also 1 and 2. And this is EIF2. And then those guys interact with this complex here. There's a complex that contains the proteins A, and G. So these guys there, it's called, big complex is called EIF4. And they don't show you that. Or EIF4F is the complex that contains all of those. I'll show you that in a minute. And those actually recognize this thing, they bind to the mRNA, and they form this intermediate complex, this 43S complex that's now has the ribosome, the messenger RNA, and the first tRNA in place. First tRNA in place actually. I'm going to show you there, but it's there. So here's the complex that forms. Here's our 7-methyl-G cap. And of those initiation factors I just showed you, those that are part of the EIF4F, the EIF4F complex can be E, A, and G. So here's A, G, and E. And you can see those three proteins interact with each other. The EIF4E is a complex or protein that has the binding protein. So it has the 7-methyl-G binding protein. EIF4 recognizes the cap of a messenger RNA. Therefore, this complex only sets up on messenger RNAs that have cap. Make sense? EIF3, remember, was bound to this ribosome here, to the small subunit, right? 1 and 3. Well, remember in prokaryotes, 1 and 3 are essentially lost when it binds. But here, EIF3 has another role. It actually helps to link this EIF4F, these 3 subunits here, to that ribosomal complex. You can see how it's in between there. So they're bound to 7 cap, and they're going to then initiate the ribosome. Once this forms, it's going to, the large subunit is going to join it, and then it's going to move on down here. The ribosome is going to release from the EIF4G and move on down to that first AUG. So there's another protein that's really not there, but it's sort of an interesting one. It's called EIF4BP. It's a binding protein. So it's a binding protein that binds to EIF4. inhibits translation. Right? It inhibits translation by binding to this. Now, when cells grow in growth factors, there's many factors in there. Many of the growth factors you learn in cell biology allow cells to grow. Growth factors, right, are why when you do tissue culture in eukaryotic cells, you use serum pretty much. You put in calf serum or serum because there's growth factors in there. And those growth factors stimulate receptors on the surface of cells that are kinase, that phosphorylate proteins, and they start phosphorylation cascades. So one of the things that growth factors do is they stimulate these kinases that phosphorylate this 4-EBP protein, right? And when it phosphorylates it, it causes a conformational change in 4-EBP, which inhibits it from binding to EIF4E. So if this guy inhibits this guy, which inhibits translation, if you phosphorylate it, so we no longer can bind to this guy, then that induces translation. And one of the things that growth factors do in tissue culture cells, right, is grow in growth factors, and they cause those cells to grow and divide. And in order to grow and divide, as you know, they have to double everything in the cell, including RNA, DNA, protein, and lipids. In order to be able to double all of their proteins, they have to turn on protein synthesis. So growth factors induce by regulating the effectiveness of the AIF4F. It's inactive because it's bound to this guy until growth factors turn it on. Does that make sense? So in cell biology, this is a very, very important thing. It turns out, I don't know that we'll talk about it, but there are many other strategies that cells or viruses use to turn down or turn on translation by messing around with the IF4F. A lot of the viruses, remember when viruses affect a cell, what they want to do is they want to make all their own proteins. They want to take all the host machinery to make more viruses. And part of that path is to shut down host processes or host synthesis. So what viruses do is they come up with an enzyme. Some viruses, not all, but some of them, SV40 does, has an enzyme that phosphorylates that inhibits the IF4E. So it inhibits translation of those proteins. And then it has developed for its own messenger RNAs, EIF4E independent translation processes. So this guy here is a very important transcription initiation or translation initiation factors. Okay. There's another protein in here, right? This is PAB1P. And you notice that this complex, this EIF4F complex, which contains these guys, the one component of it, G, is very important. G has binding sites for E, has binding sites for A, has binding sites for I. A, by the way, stabilizes this whole thing, right? Because it binds to G, but it also binds to the ribosomal subunit, so it sort of helps to stabilize this whole complex. It also has functions in attracting the large subunit of the ribosome. One of the things that G does is it binds to this protein, which is poly-A binding protein 1P, poly-A binding protein 1. And it binds to the poly-A tail. So this is the poly-A binding protein that's made in the cytosol of the cell. It binds to the tail, and it's involved in this whole process of initiation. So remember what this slide's about, right? It's the initiation of protein synthesis in eukaryotes. This is what it looks like. It makes sense. We always thought, right, that you had to initiate at the 5' end so that you can translate this coding information from 5' to 3'. You can translate it. Now here's the 3' end, that poly-A tail. It's passed. This is on the 3' untranslated region. There can be hundreds of nucleotides in here before you get to that poly-A tail. But that poly-A tail is now evolved in the initiation process. So here's another cartoon that sort of shows that happening, as well as an electron micrograph. So here's that stuff we talked about. There's the 5' cap. There's 4E that's binding to that as well as the proteins. There's G. Remember I said G binds to E. It's part of that 4F complex. It also binds to the poly-A binding protein. This green guy is PAB1A, and it's bound to the poly-A tail. So when protein synthesis initiates, you get this structure like this. And there's a lot of electron micrograph or pictures that show that structure intact. The protein synthesis begins on these round structures. Now also notice this shows you this is a polysome, we can see them in cells. Here's the cartoon one more time. And